All right, so good afternoon, everybody, and welcome to the seminar. Our speaker today is Xavier Picó, who actually made my life easier by preparing a slide to introduce himself. So without further ado, the floor is yours. Thank you.
Thank you very much. Thank you, Leticia and Dia, for being responsible of the seminars. Thank you, our communication team, Carlos and Sandra, for making these available out of these doors. And thank you very much for coming. Well, today I'm going to talk about genetic variation. But first, I'd like to introduce myself for those who don't know me. Well, I got my PhD in ecology on population dynamics and matrix population models at CREAF back in 2000. So quite soon I realized that I was not sampling individuals in populations. I was sampling something else, like genotypes, individuals that were genetically related, and that such a relationship affected the characters I was studying. So during my 3 postdoctoral fellowships, I got into population genetics, covering different aspects of population genetics, molecular biology, plant biology, till realizing that ecology and genetics are 2 sides of the same coin if we want to understand evolution and evolutionary change. So as a predoc and a postdoc, I conducted several stays in other research centers, short stays between 1 and 3 months every year. But for me, that experience was fundamental to define the mainstream of my research and the main questions that I wanted to address. Okay, well, you know that in science we normally struggle quite a lot when we pose very simple questions, right? So what's gravity? So why is the universe expanding, right? But what's a species? And where are the boundaries of a population? So these are simple questions, but with difficult answers. So let's assume for a while, perhaps during this seminar, that we are able to identify specimens and we are able to delimit the boundaries of a population. Just for a while, let's make that simple. So we can pose another simple question. How much genetic variation is in a population, right? And this is important basically because selection acts on phenotypic variation, which in turn is determined by genetic variation. So this is why this is important. Okay, well, genetic variation, you have here the bunch of processes affecting the amount of genetic variation in a population. We have drift, which is a random process, and if you gamble, you lose. So you lose genetic variation. We have selection, that also reduces the genetic variation in a population, other means, other goals. We have non-random mating, inbreeding and inbreeding depression, and bottlenecks that overall reduces genetic variation in a population. On the other side, we have mutation, that it are also random out of coding regions, of course. But non-lethal mutations tend to accumulate in populations, so they increase genetic variation. And we have migration. Migration, what it does is to bring new genetic variants into the population, which by recombination and breeding boosts genetic variation within populations. Outbreeding depression can be a problem in some particular cases, but normally it increases genetic variation. And finally, we have population expansions, which is an interesting phenomenon, because at the front of the expansion, mutations tend to accumulate. So you have the feeling that populations are increasing genetic variation in those edges. All the results I'm going to show here are based on Arabidopsis thaliana. One, in my development as a researcher, I took the decision to quit everything and focus only on Arabidopsis thaliana as a model plant. Here you can see at the tip of the pencil, I showed this picture several times, but I think it's quite showy. This is a 2-3-week-old seedling in the field. This is a 2-3-month-old rosette. probably overwintering or ready to overwintering. You can see here a headed pin. We use this to tag and monitor plants in the field. Flowering at different stages, starting, advanced flowering, and fruiting. So as you can see, Arabidopsis thaliana, the model plant, is doing the same things that any other flowering plant is doing. And probably they are using the same genetic machinery to do the same things, adjusted to its particularities, right? This is why model systems are so useful and powerful in science. Okay, so once you have an idea very clear and you know exactly what you want to do, the best thing you can do, in my opinion, is to prepare, develop the materials to work with, to answer the questions you are interested. So with my long-term collaborator, Carlos Alonso Blanco from CNV, we've been collecting Arabidopsis populations in Portugal, Spain, and Morocco for almost 20 years now. So beyond the number of populations, the interesting thing here is that we are covering a wide spectrum of environments where the species has been able to adapt. So the potential to study evolutionary change with the same organism is huge. What we do is to collect seeds. In Madrid, we multiply the seeds, we store the seeds, and we characterize phenotypes, genotypes, we sequence genomes to address the questions that we think are interesting to understand the ecological and genetic basis of evolutionary change in plants. So let's back to my original question, how much genetic variation is in a population? Well, one way to address this question is to think about the lower and upper limits of genetic variation. In one case, we can have that all individuals are identical genetically, so no genetic variation. And in the other case, all individuals are totally different and even not related genetically, which there's always some sort of relationship, but we tend to a maximum genetic diversity, in genetic variation in that population. Do we see that in the field? Well, almost. So here you can see six populations. We have the number of individuals, and we have the number of individuals with different whole genome sequences. So in Bon, Agu, and Kai, we have that between 1/4 and 1/5 of the individuals have different genotypes. So we have a low genetic variation in these populations. On the other side, we have just the opposite. Between 3/4 or 4/5 of individuals have different genotypes. So we have a lot of genetic variation in these populations. You can see that in red, these Bon and Kai, for example, they tend to be in these environmental extremes we are sampling across the region. And here, for example, POP and MDC, they are intermediate populations. So altitude could be a surrogate of the ecological driver for that. But look at Agu and Mar. They are at a similar latitude, but genetic diversity is very different. The particularity is that Agu is in a region where the species is rare, and Mar is in a region that we call Arabidopsis country. So wherever you go, you find Arabidopsis. So that could be another explanation. So my point is, can we generalize this result? Is this as simple as it seems? Well, to do this, we have to explore the distribution across space of genetic variation. You will see that I will be changing genetic diversity, genetic variation. I will be talking about genetic variation or genetic differentiation. So genetic diversity is when you handle a whole organism or a bunch of populations across a vast region. Then we are talking about genetic diversity of that organism in that region. And then we can talk also about genetic differentiation. And when we talk about genetic variation, is the genetic variation within populations, right? It's a particularity, but you will see that I'll be changing all the time depending on the example. So let's have a look at this. So in this study, we used 278 populations. We genotype with neutral SNPs, about 6 individuals per population. And what we can see here is that here in red, the number of populations with low or no genetic diversity is substantial. It's about 63, 64 out of 278. And the rest in blue, they have different levels of genetic diversity. When we look at the altitudes, we can see that populations with low or high genetic diversity tend to occur everywhere. And in the map, I think that you can see the red dots and blue dots. They occur actually across the whole place. So, well, the picture is a little bit more complicated. So the next question is, which is a simple one as well. So which are the ecological drivers of this spatial distribution of genetic variation at the regional scale? All right, so basically what we found is that there are two factors driving genetic variation. Precipitation, it's not the amount, it's the seasonality in precipitation, and pH. So what we can see here is that the spatial distribution of genetic diversity is quite heterogeneous, right? So we can see these nuclei of populations with high and low genetic diversity. In a way that if we are in a zone with a low seasonality in precipitation and low pH, no, it says high there. Yeah, perhaps it's high. Yeah, it's a weird combination. Then we are in a rich genetic area, right? So we can identify this kind of patterns, and the first message that emerges from here is that genetic diversity is rather heterogeneous across the space, right? So what about at local space? At the local scale, you see, I changed to genetic variation here. Well, and here let's rescue one of the 6 populations we've seen earlier, MDC. We've been monitoring and sampling this population since 2012. We are genotyping individuals with microsatellites. It's cost-effective, and the number of individuals it's hard to handle. And you can see that we cover, this is about 7.5 hectares. And every year we cover the population, and we sample all the individuals that we can find. We collect the seed, we store the seed, and we genotype them, right? The results are quite similar in terms of how heterogeneous genetic variation is within a population. In this case, this is a surface map of genetic rarity, which is kind of surrogate of, it gives us clues, hints about how genetic diversity is distributed within the population. And we can see the same nuclei of regions within the population with high and low genetic rarity in this case. Okay, let's pose the same question. Which are the ecological drivers of a spatial distribution of genetic variation within a population, which gets closer to the original question of this seminar? Yep, I don't know. We tried, we tried, but I can give an answer, just a mere speculation, which I don't like very much. So I have some homework to do, which is to identify the local scale drivers of genetic variation. I can tell you something. We identified spatial heterogeneity, environmental heterogeneity, using vegetation types. So genotype growing in scrubland, genotype growing under a holm oak patch, dry riverbed, grassland, but it's not enough. So probably what I have to do is to look at the close vicinity of genotypes and see what's right around each of the plants. So I have to scale down the intensity in which I'm estimating spatial heterogeneity. Okay, now think about it. When we go out to the field and we collect samples, right? And these samples, we move to the lab and we genotype these samples and we estimate our parameters. What we are doing is to take a picture, a static picture of a population that it's not static by no means. So populations are dynamic, they change, They have problems in which genetic variation can increase, decrease, can occur many things. Obviously, we have the means, the tools to infer what's going on. But is that enough to understand what's going on in a population, or to try to answer our question, how much genetic variation exists in a population? So this is important to realize that in population genetics, in population biology, we have the temporal dimension of the whole thing, which is as important as the spatial dimension. It's easy to sample across the space. It takes time to sample over time, right? So what did we do to address this question? So we developed what it's called a resurrection approach, which basically it means that in this case, for example, we sampled 4 populations in 2003, and 10 years later, we resampled the same populations, the same patch, exactly the very same protocols applied to the 2 samplings. We estimated genetic variation in flowering time in a common garden experiment. We genotyped individuals, in this case the ancestors, and after several generations the descendants. So we have a real-time change in these particular populations. And because this is Arabidopsis, and we can get to the gene level candidate genes with tested function, we sequenced the coding regions of 2 flowering genes, FLC, a central gene in the vernalization pathway, and Fry, its main regulator, and DOG1, the most important gene of the dog family, affecting seed dormancy, which eventually determines germination time, right? So what we found? Look, genetically, in just 10 years, populations got differentiated between 20 and 34%. In this graph, this is an example of one of these populations. You can see the hollow dots, the ancestor population, and the field dots, the descendant population. And you can see how in 10 years, the same population got differentiated over time. And genetic variation in flowering time, well, we found different results. One population, on average, advanced flowering, nearly 3 days, and the other population delayed flowering, almost a week, right? So ecological systems, biological systems are dynamic over time, and we have to consider that. I don't show you the data from nucleotide variation in genes because we didn't find any significant difference in functional polymorphisms affecting gene function. So no changes at that level. Which are the ecological drivers of this temporal, so evident temporal variation within populations? Yeah, sorry, but I don't know. I have to be frank here. So more homework to do, which are the regional scale drivers of temporal genetic diversity. Here the problem was that statistically speaking, my unit was population. I have 4 populations in 2 times, not enough to extract conclusions. There's a trend, you know, some populations got drier environments, some others were wetter, you know. In all of them, in 10 years, temperatures got high, right? And there's a trend that it seems that if you go drier, you are losing more genetic variation, but it's just a trend, but not enough to get somewhere. So in my previous project, the goal, actually the project was called Resurrect, because the goal was to sample different populations in Iberia and Morocco over almost 15, 20 years in difference. But we got the lockdowns, pandemics, blah, 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 you know. So I had to reschedule everything. I know I'll do it, but resurrection experiments imply a ton of work. So, and we handled 4 populations, and that was a serious business. So, but I think that 20 populations, well-selected 20 populations in the right environments could make the thing viable. And now the remaining step is the same question. What's the extent of temporal variation in genetic variation at the local scale? So back to MDC, remember that we've been sampling this population for 10 years in a row. So here you can see for each of the years from which we have now genetic data in orange. You see MLG means multilocus genotype, but it's a genotype, right? So in orange, you can see the genotypes represented by one individual found in 1 year. So unique genotypes. And in red, you can see genotypes for which we have more than one individual. Actually, we have multiple individuals. Normally, we observe them in different years, so common genotypes. The striking thing here is that every year we observed between 40 and 75% of unique genotypes. So, what's the amount of genetic diversity in a population? A lot, huge. So I have to ask it, which are the ecological drivers of this temporal variation of genetic variation at the local scale? Don't panic, I get something. Right, okay. This is a graph showing the fine scale genetic structure for each year. And you can see that at small distances, we have a significant pattern of genetic structure. So basically what we are sampling here is a population with a bunch of genotypes that create like small clusters, like families. So every year we are sampling individuals from different families. The point is that there's a lot of these genotypes because every year, we don't see an increasing number of genotypes previously detected in previous years, right? So I must confess that I'm partially satisfied with this explanation. So we'll keep sampling MDC. So that's this is a fact because the more we sample MDC, so the more questions emerge, and the more interesting the whole thing gets. Okay, earlier I said that genetic variation is important because selection acts on phenotypes that are, of course, affected by genetic variation. But let's develop this a little bit further. So what's the value of genetic variation? Let's go back to MDC. Actually, MDC, I would say, is the jewel of the crown, right? So, we not only genotype individuals, but we phenotype them. So we estimate genetic variation in life history traits and fitness in common garden experiments, as you can see here. These are the results of the effects of ecological and genetic factors on genetic differentiation in key life history traits and fitness components, like germination, flowering, and fecundity, right? What we can see here is that ecological factors, isolation by distance, it's basically the result of dispersal limitation. So the far away you are from your neighbor, the more different you get. And vegetation type, as explained earlier. And you can see that the similarity in these ecological factors do not explain differentiation in life history traits and fitness components. In contrast, genetic factors like the extent of relatedness between individual pairs or the degree of genetic rarity do explain genetic differentiation in traits, right? So it's clear that the genetic composition of individuals matters a lot to understand what we observe in the field. Because flowering time is the major target of selection, we identify also isolation by distance, which is a normal result, and fecundity has a very strong environmental component. So not even the genetic relatedness can explain differences in fecundity, right? So do we see, this is at the local scale, I start in the other way around. So can we see this at the global scale? So we pose the same question, and this is Bernardo's second chapter of his thesis, co-supervised by my other long-term collaborator, Arnaud Marcet from CREAF. And here we use not only our collection, but other accession collections from other labs of the Arabidopsis community. The results are practically the same. So the contribution to phenotypic differentiation for different traits is mainly driven by genomic relatedness among accession pairs. You can see that this applies to structural traits, plant height, for example, chemical, basically plant defenses, development, life history traits and fitness, and response to biotic and biotic stresses. But you can see that genomic relatedness explains phenotypic variation at this larger scale. Then isolation by distance, it's a matter of dispersal limitation. And finally, habitat suitability, which is our factor more related to the extent of local adaptation of these populations, right? So another reason to understand why genetics are so important to understand evolutionary patterns. All right, so I posed simple questions, and I think that deserves simple conclusions, right? So I would say that when you go out to the field and you collect your samples to do whatever you want to do with your study species, genotype everything, because that can explain a lot of the patterns that you observe out there. This is the current list of major collaborators, PhD students, technicians, postdoctorals, PIs, and well, more than collaborations, I have the pleasure to work with good friends of mine, which is always a privilege. Thank you very much.
Thank you, and we have a lot of time for questions, and I also want to remind those that are watching us on YouTube that they can use the chat box for their questions.
I always make questions, so now it's time for revenge.
I have a very, very basic question about when you say same genotype, do you mean like a clone? Or are you talking about just very closely related individuals that you can't tell apart with your marker set?
Right. Well, with the set of markers we used, and we had a Driscoll to assign two individuals to the same genotype, because we were considering the genotyping error in these markers. Mm-hmm. So with the markers that we were using, we can say that these two individuals share the same genotype. They are not 100% identical, right? But the same happens with whole genome sequences. So after applying your criteria, then you can group the individuals, right?
Okay, and you were using two different marker sets in different things, I think, right? SNPs sometimes and microsatellites sometimes?
Yes. We started with microsatellites because they are cost effective, and they are very informative, I have to say. Yeah. So you don't need whole genome
and they don't have so
sequences
much
for everything.
Bias.
Then we developed a set of SNPs across the 5 chromosomes of Arabidopsis, and now it's cost effective as well to get whole genome sequences. So yeah.
So your SNPs, you developed them for Iberia, or what? What is the target population?
We had 3 pools of SNPs to avoid the ascertainment bias, right? And eventually we pooled them all together because bias was ridiculous. Okay. So yeah.
So one more question, and then how big is your SNP panel?
Excuse me?
Your SNP panel, how big is it? How many different SNPs is in
Well,
your
now we are using about 1.8 million SNPs.
Oh, okay.
And 96% of the coding genes have at least one informative SNP in it.
Okay.
Right? Yeah. But it depends on the question. So, you know, for population genetics, genomic tools are powerful, are nice, but to get the first picture, you know, actually we are using SNPs because we can genotype every year 150, almost, we never reach 200 individuals, but 170 individuals every year. But then we are using the panel of individuals genotyped with the SNPs to identify those individuals for which we get the whole genome sequence, right? So it's a tool to redesign experiments.
Okay, one more question then. How big is the genome?
It's about 250, 260 megabases, right, Fede? No. More or less. So, yeah.
Awesome.
Yeah, you can handle it. Yeah. Yeah. No polyploids, no weird stuff, so that's nice. All these populations, they have 5 chromosomes, which you know, which is useful.
Can I? Okay, I have a question as well. It's related to the ecological factors that dictate patterns of diversity,
Yep.
because you are talking about humidity and so on, but there are other biological factors like population density. Do you have any differences? Because I think, to me, it's the first factor that would come to my mind. And differences in the population density, the effective population size is locally larger, and that would increase the genetic variation. Do you have any way to control for that?
Yeah, sure, yes, we do. Actually, the 6 populations we've seen earlier, they also differ in that kind of patterns. So this is why I said let's assume that we can define the limits of a population, because a population of Arabidopsis actually it's a group of patches across the area. You know, in some cases you have a bunch of individuals accumulating in good sites, good spots, and normally those individuals are more genetically related among them than others. In other populations, you know, what you have is almost a carpet of individuals. There are individuals everywhere, and all these kind of things are affecting the amount of genetic variation. So what we are doing is to sample the same area. We are mapping everything to have the idea about the population structure that is affecting these parameters. But all these populations, we know that they are old. We have historical data to make sure that there were not major disturbances affecting the genetic variation. And they are large. So there's a bunch of individuals in all of them. In some of them more than in others. So, and now we are estimating population size. So we conducted a seed bank experiment because we go to the field and we collect seeds from reproductive plants. But the large part of the major part of the population is under the soil. So we have some estimations about the population size considering the soils buried in the soil seed bank, right?
So, wait, sorry.
Thank you for the talk. I want to make also some questions to understand better the system but from the ecological point of view. So I'm assuming that dispersal distance is small, sorry.
Mm-hmm.
And I'm also assuming there is a large seed bank.
Mm-hmm.
So for example, for your temporal analysis, to which degree you are just seeing different bed hedging strategies for like different genotypes emerge depending on the temperatures. So the overall variability is quite homogeneous. It's just you detect different spikes of this variability.
Yeah, good questions. Well, we have estimated that the maximum, or at least the maximum dispersal that we have estimated with genetic markers, it's 11 kilometers. But that's unusual, and it's a huge distance. So normally each population has unique genotypes that differ from the other populations. The difference is in the genetic relatedness between populations. But it's clear that the species is able to change the genetic composition, right? And probably through rapid evolution and local adaptation, in a few generations, you have a unique genetic makeup for these populations. The other important thing is that our estimated half-life span of seeds in the soil, it's about 3 years. So in 10 years, we make sure that the generations sampled in the first year are already different from the other, right? And about the bed hedging strategy, that's a more complicated thing, because for a long time people thought that Arabidopsis had two strategies: winter annuals, so I germinate in fall, I overwinter, and I reproduce in spring, and spring annuals. So I remain in the soil seed bank, and in the spring I germinate, grow, reproduce, and go back to the soil seed bank. Well, our data is very clear on that. We have everything in all populations, regardless the environment. So actually what we are seeing is a huge plasticity in life history, actually because it's the best strategy you can do. Why bet for one strategy when you can keep both, right? There's a trend, so that's a preference for lifestyle depending on the environment, but eventually you can see everything everywhere, which is quite remarkable.
So I see a couple of questions.
Thanks. I'm glad that the word dispersal has risen, because surprised you didn't mention that. So, I mean, I expect, part of my question, for a plan like this, really, that our understanding of real, true dispersal processes in the field is extremely low. And I think we're dominated by this flawed paradigm where we kind of predict dispersal mechanisms just looking at the seed morphology. I really think that's not a good way, not a good approach. And I wonder whether, with your data, you can really get some better insights into seed shadows and spatial variation in seed shadows, because I imagine even on a local scale there will be tremendous variation in seed dispersal distance between patches.
Yeah. Well, at our scale, at a human scale, it seems that the species does nothing, but the species is everywhere. So it reached all kind of environments and had time, basically because I think that there's rapid evolution going on. It had time to create, to thrive in all these kind of environments. But it's difficult to identify these dispersal kernels, basically because we should genotype a bunch of individuals across huge areas. And now what we are doing is to increase the density of populations to detect, and the first time we detect the same genotypes in two different populations was in one of these studies that I showed, in which we found because for decades people are finding different genotypes in different populations. So what you are finding is that close by populations are quite similar genetically. They are already different, but they are quite similar, meaning that they are related, but they had time to differentiate in a very short period of time. So that's why it seems that dispersal is not playing a role. It's important because the plant is everywhere, but the local adaptation, the rapid evolution is so fast that you get a strong differentiation in a very short period of time. You know, that's the complexity of the whole thing.
Right, but you said that you could not explain the local spatial variation, right, within a patch.
Not yet.
So at that scale, within that site, I expect there'll be huge variation in dispersal distance.
Well,
and so according to
a few meters. We see these genotypes, common genotypes with more than one individual that we detect in different years. Sometimes, you know, they are in different places of the population. So we have wild boar digging out the soil. We have burrows for shelter, you know, these kind of things. So eventually, you know, the population is mixing up in a way, right? But to detect these patterns, actually we have to increase sampling size at very short spatial scales to catch these dispersal kernels. That's what I think needs to be done.
Yeah, okay, that's great. I mean, I like the way you mentioned the boars, because that would be an example of something which could be really important but isn't recognized by the dispersal syndrome paradigm.
Oh, well,
So-
I think that in this case they play a role, that's for sure.
Yeah,
Yeah.
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 Thank you. Is it directly in response? If not, I had Maria before. Well, okay. Was it about this person?
No, it's not this person.
Okay, so sorry.
Forget dispersal. I wanted to ask something regarding the previous discussion you had about life history traits and their and how variable they are. And when I heard you respond to that, my immediate thought was, oh, is there strong density dependence in the population? So is there other feedbacks between population density or density of the neighbors on the evolution of different, the change of different genotypes, the life history traits and so forth? Is this something you've looked at?
Well, I don't know. I don't know, because now one of my short-term goals actually is to characterize the micro scale at which Arabidopsis like this one is developing, is completing the life cycle. So in these cases the plant was quite alone, but in some others the neighbors of this plant can be any other plants. So I don't have a clear picture about what really matters at this scale to affect the fitness of individual plants at this scale. But I know that I have to narrow down quite a lot to get that picture. So it's a complicated, but the new project proposal I'm working on right now, that's one of the objectives. So yeah, but I understood, that the I told Raul when he got here is imagine that this is mini pines, but it's the same thing, because he's a forestry ecologist. So, but obviously, everything is tiny in Arabidopsis, so it means that I have to just narrow down the scale at which I extract data from the field.
Yeah. Hola, Xavi. So regarding your local experiment, in which you have sampled so many individuals during 10 years, I guess you have more spatial genetic variability than year-to-year temporal variability.
No.
You have more year-to-year?
Similar.
Similar?
Similar. No difference at all. If you explore spatial patterns in genetic distribution or temporal patterns in genetic. That was a surprise, actually.
Okay.
Except the fine scale genetic structure, which is quite clear. That at short, at the small distances, what you have is relatives growing together, of course.
Okay, because my question was that it doesn't have any sense now, but if you have tried to estimate how much time do you need to temporal variation to overcome spatial variation. Yeah. And but since you have told me that 1 year is enough, is already answered.
Well, I don't know how to answer that question. How much time do you need?
I mean, for example, I imagine that you can make like a similar analysis of the analysis you have done of spatial genetic structure, in which you compare the kinship between individuals across spatial scale. You could do the same, comparing kinship between individuals across temporal scales, and see to what extent, how much time do you need to lose the temporal autocorrelation.
Yeah, that's a good point. The problem that we have is that every year we are observing a huge amount of new genetic variation. Every year, you know, that's a problem. So I was expecting that after 10 years, you know, most of the genotypes were already seen before, but it's not the case. So, you know, there could be perhaps a fast local extinction of genotypes, you know, and new genotypes are appearing, or our estimates of the number of seeds per square meter are huge. So if that's true, it means that there's a huge amount of genetic variation waiting for an opportunity to show up, which is, I think, what's going on here, you know.
Okay. Thank you.
Thank you. I saw we had a question in the front, but I also wanted to read you some of the questions that we've had online.
Oh,
so
excellent.
I'll start with Joaquin Ortego Lozano's question. So, small question. Could genetic differentiation through time be simply explained by genetic drift? I guess that populations are pretty small, and so they could experience a lot of genetic drift and quickly differentiate through time.
Well, drift is omnipresent. Even in large populations, we know that drift are changing the genetic composition of populations. But these populations are not small. That's the difference with Joaquin's remark, right? So yes, drift is like a basal noise. You know, it's always there, always there. So I don't think that drift is really doing something, but I don't have data to assure that.
Is it?
Yeah. Have you estimated the effective populations? Have you estimated the effective population size of some of these populations that you have the whole genomes for?
No, because I have troubles in estimating population size. I estimated the effective population size, right? And yeah, but it's related. So populations with a low effective population size have low genetic diversity. So. Well, it depends, because my estimations are at the patch level and not for these 7.5 hectares. But, oh, wait a minute, in the resurrection experiment, we estimated the effective population size. It was around of dozens of individuals only. So it's not about 1,000 or 3,000. We are talking about 20, 30, 40 individuals. I have to check that out, but more or less.
Another online question from Miguel Fortuna.
Okay.
Do you think herbivores, for instance insects, might play an important role in shaping the genetic variation among populations?
That's a good point, and that's another objective of the new project. So Nacho helped me out with that. And it's interesting because for some reason insects ignore rosettes. They wait the plant to flower, basically because when these plants flower, all the resources go to the flowering buds, and they are waiting for that rich material. And in some cases I remember a population up in the Pyrenees. There were different patches, and half of the patches of the population went extinct because herbivory. Every year I got there and I saw the stalks cut off. Every year. After 3 years, that patch was gone, right? Which fits with the estimations of half-life of the half-life span of seeds in the soil seed bank, right? So these populations actually need to replenish the soil seed bank every generation. They have, you know, they have 2 or 3 years, but if not, you can get in trouble.
So there was a question here.
Okay.
Thank you, Xavi, for this interesting talk. Before doing my question, I need some help to shed some light to this plant system.
Okay.
Arabidopsis is autogamous, right?
Right.
So self-pollinates.
Right.
And have in a 90%, right? And tends to be homozygous for the alleles. All the seeds from the same plant are the same genotype and are the same genotype as the parent in your molecular analysis?
I guess so.
So, part of your results, you show that there are a lot of unique genotypes. This means that from every plant, maybe just one seed recruits.
Well, no, we don't estimate that in that way because, as you know, these populations have between 5 and 10% of heterozygosity. So, meaning that there are outcrossing, which is a huge force boosting genetic variation, right? So the way to genotype these individuals is not by collecting this rosette in genotype. We collect the seeds, we grow the full seeds together, and we sample the descendants, and we pull material from 6 plants from each maternal line. So if the mother plant is heterozygous with 6 individuals, 6 full seeds, we are catching as well the heterozygosity of the mother plant. Some of them will be homozygous, some of them will be heterozygous because they are segregating, right? So this is the way we have to estimate the genotype of this plant. So I would wait this plant to shed seed, I would collect the seed, then we grow the descendants, the full seeds, and then genotype a subsample of them.
Here it comes the question. Taking into account this dispersal limitation, taking into account how many unique genotypes there are in the population, it looks like there's a lot of within genotype competition. What do you think about that? The genotypes are competing within.
What do you mean by that?
From all the seeds from the same plant, the most competition is happening within the seeds from the same plant.
Yeah,
there's a lot of competition within genotypes. That is very no intuitive, right?
Well, no, but the way these plants work is the following. So if a flowering plant gets in trouble because of a drought or whatever the question, what these plants do is to assign all the resources to the fruits developed and then stop the development of the rest. So what you see is a bunch of abortion in the flowering stalk because all the resources, so what they are doing is to assign the resources available to a small number of seeds, but those seeds are viable, right? So yeah, that within genotype competition, yeah, I don't know, yeah.
Thank you. So we still have 2 questions online and a couple of minutes left. I don't know if there's any more questions here in the room. Okay. One. Well.
Yeah. Thank you. So thank you for the talk. I was surprised with the turnover of genotypes in a single population. And for me, that could explain why you don't find a spatial pattern to explain variability, because if it changes from year to year,
yeah, I agree.
And you have a map that I guess that is the summary, no?
I agree. Yeah.
So rarity is somewhere, but the rarity every year is in different places probably. Yeah. So that's a hard work, no?
Well,
how to characterize that.
Yeah, for different reasons. These are the ideas now for the new project proposal. But probably I will change the way to sample and genotype this population. So perhaps now I will select different patches and genotype everything within those patches, right? And I think that could be a better option because I'm working like this is a scrap, you know, a rosmarinus, for example, and it's not the scale at which you have to work with this plant. It's much smaller.
It's millimeters.
Exactly. Yeah, took me some time to understand that.
So next we have a question from Christoph. Thanks, Xavier, for sharing your work. What happens in terms of genetic diversity at the peripheries of species distributions in your system?
Okay, yeah. Well, not much. Because, for example, if we think about an edge population in Spain, for example, we can go to Valencia, to Murcia, you know, arid places, but the plant knows it's not growing in arid places. It looks for that small patch with humidity, you know, in the right aspect, you know, perhaps a small oak forest, and the plant is there, surviving there, right? But the genetic diversity that we find there, or the phenotypes that we find there, you know, it's more at which altitude are you sampling, whatever the place. It doesn't matter really the area, but the altitude is an important surrogate for plant performance, to predict plant performance. So when in those edges are important, but those edges are not specific adaptations in my opinion, but remainings of former larger populations perhaps, which is a different thing, you know.
Thank you. And last question online by Enrico Pasicalupo. Thank you, Xavi, for the interesting talk. I have a question. When exploring the effect on phenotypic traits, what is the conceptual difference between genetic relatedness and isolation by distance? I'm not sure I understand correctly, but wouldn't high effects of genetic relatedness translate into high effects of isolation by distance, as further away individuals have lower genetic relatedness?
Yeah, well, Enrico, you're right, because isolation by distance and genetic relatedness are always, they have some sort of relation. But genetic relatedness, in this case, we don't care about the position of individuals, because we can have the same, and we have the same genotype in different places within the population. The important thing is that whatever the distance between you and me, if we are similar, we'll perform similar in the common garden experiment, right? That's the particularity of genetic relatedness. And isolation by distance, what really matters is how close we are, independing whatever the genetic relationship we have, right? But both factors are related, yeah, pretty much, yeah.
Thank you. So I'm turning now to the audience, if you have any last questions or comments. And if not, I think we can thank again Xavi for
thank you for
this
the questions.
presentation.
Yeah, thank you very much.